Greetings, my dear friends, and welcome to yet another Reading Epic Threads live stream. I'm your host, Patrick Gunnels, joining you from Houston, Texas, this July 22nd, 2022 at 3:32 p.m. Central Daylight Time. And it's a very special episode of Reading Epic Threads today because we have a guest, Steve Kirsch, who is going to join us for an informal discussion about what the content of a debate would be, talking about the existence of SARS-CoV-2, the existence of pathogenic viruses in general, and just basically how the debate would unfold. So without further ado, I would like to bring in our guest today, Steve Kirsch. Steve, how are you, sir?
Good.
So what we began with when we decided to talk about the existence of pathogenic viruses was there was a challenge issued by some of what we call Team No Virus, where they put together a challenge to laboratories to replicate the finding of an alleged pathogen.
Great. So would you be participating in this debate?
Absolutely.
Awesome. My first question is, how did you acquire your knowledge of virology?
I just by Dr. Cowan, Dr. Kaufman, Sam Bailey, Mark Bailey, Robert O. Young.
Okay. Did you ever read any traditional virology textbooks or ever study it in school?
No.
Okay. I just, I'm not trying to judge people. And believe me, if you don't have I judge people based on their knowledge and not on what their credentials are.
Okay? Respect.
All right. So what is the definition? We're talking about, is there a virus or not? So it's really important to start off with a definition that we can both agree on, right? Because we can't argue on, is there a virus, if my definition of a virus or someone else's definition of a virus is not the same as your definition of a virus. Because if the definitions are different, then you could be correct. Yes, a virus doesn't exist per your definition. So we really need to start with an mutually agreed upon definition of what a virus is.
I agree.
Okay, great. So do you think that we have that in the Sam Bailey challenge, a mutually agreed upon definition of what a virus is?
I'll give you my definition of a virus, and then you can tell me what you think.
Well, no. Could you answer my question first before you do that? In Sam Bailey's statement about challenging the existence of a virus, do you believe that she explained the definition of a virus that mainstream virologists would say, yes, that is what we're talking about here?
Well, the description given is a replication competent particle that uses the internal machinery of a human being's cell to cause disease.
Okay.
Is that acceptable?
I think that's acceptable.
Good.
So the next question is, do they exist? Do viruses exist? And is your point that the measles virus doesn't exist, that the influenza virus does not exist, that any of these other, the measles, I'm not sure if I said that before, the measles virus, does that exist? Do any of these viruses exist, or is virology just junk science?
Those viruses do not exist. Virology is junk science.
Okay. So then the next question is what is the accepted test to establish that a particle is a virus? What is the accepted test by virologists for determining whether something is a virus or a bacteria or a fungus or whatever? There has to be a test that people use to say, oh, we know because we do these three things, right? We've talked about what the properties are, and we agree on that it's it is not a living organism. It is basically there is RNA material or DNA material inside the capsid, which is then delivered into the cells. In the case of an RNA virus, it is then that material is then used by the ribosome of the host cell to replicate itself and express proteins on the surface of the cell and so forth, and to then create replicas of the RNA and spread it around. So we all agree on that, right? That's what the mainstream virologists believe. Or the definition of a
virus
doesn't
and the
exist.
method by which it causes disease.
Okay.
I think we've already covered that.
Right. We covered that. Okay. So what is the accepted test then? We have this understanding of what we think it is. What is the accepted test that a virologist would use to say it? It's like the duck test. Does it walk like a duck? Does it quack like a duck? Then it must be a duck, right? There may be three things on the duck test, but what is the duck test then for a virus that a virologist would use? Would he ask, hey, does this exist independently, or does it only exist through this? Is this a living organism? They would probably ask that question first. They would probably ask questions about the size. So my question is, what tests would a virologist use to differentiate a virus versus a bacteria versus a fungus? How would they characterize and say that this is a virus?
Well, you're asking somebody who has said that virology is junk science. So I would request that you rephrase it as, what test would a legitimate scientist do in order to find and characterize a virus? Would that be acceptable given where I'm coming from?
Well, let's see what your answer to that question is. Okay. I'll give you the benefit of the doubt, and we'll see whether we can agree that that's a fair test.
A fair test to determine whether or not a hypothetical particle exists and is causing disease would be to find it. And you need to find it in the fluid or tissue of somebody with the disease that you think is being caused by said particle.
Okay.
You have to do that by extracting the fluid from the human being who is exhibiting disease. And then from that fluid, you would have to extract a sample of that hypothetical particle. Then that particle would have to be examined, characterized, and then it would have to be introduced into another human being. And without any other thing being introduced into that other human being, would have to be shown to cause the same disease that was observed in the initial patient. And you would have to do that with an acceptable sample size of patients in the first case and in the latter case.
Seems easy enough. Why hasn't that been done? Why do you think that hasn't been done?
Because virology, well, hang on. I can't read the minds of people. What I can tell you is that it hasn't been done. I could speculate all day as to why it hasn't been done.
Okay, but let's break it down then, because you mentioned, and I wrote this down, 5 things that have to happen, and you say it hasn't been done. By the way, I have a hard stop at 3 p.m., which is in an hour. It's 1:44 p.m. Right now. I just want to let you know that up front. I'm not trying to get out of this. I have to pick up my wife at the airport. She's coming in from Scotland, so
I'm sure we have plenty of time.
Okay, awesome. Great. I just wanted to make sure you didn't think I was ducking out at 3 p.m. because things were getting uncomfortable for me.
Well, this is just a question and answer session. So
Exactly. Good. Nice, friendly. We're having a nice, friendly discussion.
Mm-hmm.
Awesome. Just the way I like it. Okay. So you mentioned 5 things. Find it in the fluid or tissues, extract the fluid, extract a sample of the particle, examine the characterized particle. That's what I wrote in my notes. I'm not sure. Can you elaborate? Examine the characterized particle?
Observe it, look at it.
Like under an electron microscope?
I guess. But the problem with electron microscopes is that they just crank out artifacts like crazy. But
okay, but we call it Sanger sequencing.
What you could use, not sequencing. No, you'd have to identify and observe an intact sample of the organism.
Identify and observe.
Characterize. And then as far as characterize, you observe it, you look at it, you determine what it's made out of. And then, you can get to sequencing later, but just finding an intact virion would be a very important step in the scientific method.
Okay. And when do we do the sequencing?
I don't see that there's any way to get to the sequencing until you've identified an intact organism.
Yeah, no, but you identify the intact organism, would you sequence it? Because
I
you'd want
mean,
to sequence it when you reintroduce it into someone, you'd want to sequence it first, right?
why?
And then because you can then compare the sequence to make sure that the person has the same disease as the virus, because the virus is defined by it's inside the uh, 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 um, per- inside the capsid is the RNA material. That the RNA, the sequencing of the RNA defines the virus, that particular virus, right?
A simple observation under a microscope, making sure that the particles have similar morphology, similar composition, would be sufficient to me. If you want to sequence it, that's up to you, but I'm not even going to put that standard in there because they haven't even bothered to isolate, purify, and then introduce them into healthy patients. So
Okay.
simply isolating, purifying, determining identical morphology from each individual virion, and then showing that that particle is replication competent in another patient and causes disease. All of that would have to be observed in order for the scientific method to have been satisfied.
Okay, but you would agree that sequencing, like Sanger sequencing, would be a way to sufficiently identify the virion, right?
Only if you have an intact sample of the viruses already before you use anything like Sanger sequencing or deep metagenomic sequencing or any of these methods that virologists use. None of them would be valid except in the presence of an intact virion, a sample of intact viruses.
Well, Sanger sequencing relies on pre-processing the sample, and
it's a sample from a sick person. It's not a sample that has been shown to contain intact viruses.
Not a sample that. So you don't believe that Sanger? So you don't believe that Sanger sequencing is the gold standard for identifying a virus? That RNA sequencing.
You use the word believe. This is not about my beliefs. This is about the scientific method.
Yes,
in order to sequence any organism, an identified, purified, isolated example of the organism must be obtained first. You can't assemble it using sequencing from a sick person and say, well, this is the genome of this virus because we just assembled it in a computer. And that's probably taking it to the logical next step, which is what the sequencing really does. Now, I know that different sequencing methods can sequence larger numbers of, bigger and bigger contigs and all that stuff, but none of that matters if you don't have an identified example of the hypothetical pathogen. And we haven't had that yet.
So now, of the 6 things that you talked about before, which I enumerated before, which of those are the ones that you think are not satisfied today by virologists?
None of them.
None of them.
Mm-hmm.
Oh, well, okay. So I'm legitimately surprised by that. I thought you'd say, well, they haven't done the isolation piece, or they haven't done the.
Well, let's list them off. Maybe I forgot about one that they have done. So can you
find it? Okay, so we'll take them one at a time because I'm not trying to trap you or anything.
This is fine.
Okay, so find it in fluid or tissues.
Haven't done that.
No. Have not done that. Okay.
Correct.
Okay. Extract the fluid.
Oh,
they can't find it. How can they extract it? Right?
You mean just extracting somebody's fluid? Yeah, they've done that.
Okay, they've done that.
They put tubes down people's lungs and grabbed
right. I mean, I do it all the time with the PCR test or the home antigen test.
Taking a fluid sample or a tissue sample, virologists have definitely done. So you can put a check mark next to that one.
Okay. Extract a sample of the particle.
Yeah, they have not done that.
They have not done that.
Not from the fluid.
Okay. Now, by extract a sample of the particle, do you mean like an isolated particle that you can take? Like he's just he's a one-off that that here's the particle, and put it in a test tube or whatever, and it's just one particle in a test tube. Like,
no, I'll give you an example of what I mean. I mean ultracentrifugation, density gradient, put a pipette in there, you get to the band on the centrifuge that contains only that particle and pull that out. So not, of course, one off, but a whole big chunk of only that particle to see if it is a virus.
Okay. Now, do you believe that those things, that those particles, that you do your ultracentrifuge, which I agree is one of the techniques that can be used to extract the virus from everything that's surrounding us? The centrifuge is a good way because the virus is so small and so tiny that it's a way to separate it out. But you may not just get the coronavirus. You may get, I could be infected with 4 other viruses, and I may be infected with 3 variants of the coronavirus, right?
I don't accept that anybody can be infected with something that doesn't exist.
Okay, but we haven't. Well, then you can't extract a sample of the particle if you believe that the particle doesn't exist. So let's postulate for a second that the particle exists, because that's what we're trying to show. When I extracted the particle and I give you the extracted particles, I'm giving you something in there, right?
Okay.
And you're claiming that these, a virus. There are 2 kinds of viruses essentially. Viruses that have an envelope and viruses that are naked, right?
I don't think either one of those exist. I know that virologists say that stuff.
Right. But just because you believe they don't exist doesn't mean that they don't exist, right? I could say that God doesn't exist. I don't believe that God exists because I've never seen proof of God existing. Other people believe that God does exist. So conversely, you're saying that you don't believe that the virus exists just seems like having a closed mind to the possibility that the virus can exist, because the whole point about doing this is to show that what the virologists say is not self-consistent, right? I mean, basically, you have to break down, you have to prove that there's fraud by these virologists in some way other than saying, well, I don't believe that there's a particle, and so you can't extract it.
I don't have to prove that there's fraud. I have only to critique the scientific evidence and show that it does not conform to the scientific method. And if it does not conform to the scientific method, then the existence of such particles is unproven.
Is there, maybe, but maybe not. Is there a hole in to me, as a person who I claim I'm not an expert in virology, never been formally trained in virology, but what I know about virology from my friends who are virologists and my friends who do sequencing sounds self-consistent to me, right? It sounds that the whole thing makes sense. They can look at these things under an electron microscope. They can sequence them with Sanger sequencing. There are sequences of the RNA that have been published by multiple people. And there are multiple sequences and there are multiple variants of the virus. Everything to me seems self-consistent. So what was the big hole in the self-consistency? Like, can you point out well, it can't be a virus because vaccines don't work. But in fact, vaccines which introduce an antigen provide immunity. So if viruses don't exist, for example, you could say, oh, well, the fact that no vaccine has ever been developed would be proof that viruses don't exist, because if they did exist, we would be, if viruses did not exist, then we would never be able to create a vaccine which prevented people from getting the virus. And yet we are able to.
You mean people getting sick?
I'm saying I'm trying to get to the self-consistency that the point is that I don't see anything in virology and in all this theory and in the gene sequencing that says, oh, red flag here. They were never able to sequence the viral RNA of this pathogen and find it in multiple people. They're always going to be variations in that sequencing. So you shouldn't expect to find an identical sequence in various people. But the point is that their stuff is self-consistent, right? That the viral theory of
Steve, you're kind of scattergunning with a lot of different things right now. Do you mind if we address them one by one?
No, I'm just asking you, is there something that was a red flag to you in the theory of viruses that said, oh, this cannot be true what they say because blah?
May I?
Yeah, absolutely.
Because the so-called virus has never been found in the fluid of any sick person ever. That's a big, big red flag.
Because the virus has never been found. Okay, so my virologist friends would disagree with you. They would say that it has been sequenced, that they know what the viral genome is. And when I go on the ATCC website, for example, I can find lots of these things that I can purchase. I find references to the peer-reviewed literature on this. I look at the viral sequences, which I can find. By the way, do you know what the supposed length of the non-existing virus is that they claim?
Which one?
SARS-CoV-2.
I think it's 29,000 base pairs.
Correct. Yeah, 29,811, give or take. Okay, good. Yeah. Okay. So you at least understand what their claims are.
I've looked at their claims.
Good. All right. That's good. I mean, I'm happy with that.
Okay.
That you know that, right? A lot of people wouldn't know that, so I'm happy that you know that.
Well, I'm happy that you're happy.
Yeah. Great. So, yeah. Let me see. Getting back. So the big issue, the big disconnect for you is that the virus is never found in every sick person. And I wrote every something, and then my notes dropped off there. But what did you say?
I said that just give you a big example of a red flag that starts you off to think we're dealing with something that's non-scientific.
Yep. Is
the particle has never been found in the fluid of any sick person ever.
Oh, ever. I sort of wrote every. Okay, great. Okay, this is super helpful now because we can then focus on this sort of red flag that led you to all the other things, right? Because I would contend that Koch's postulates were created a long time ago, and the virologists have moved on from that, and Koch's postulates essentially get superseded by gene sequencing.
You can't sequence something you haven't found.
Yes. Well, I'm trying to zero in on that part.
The
particle was never found in any.
You can't supersede the scientific method. Then you get into pseudoscience.
Okay.
And until you find it first,
yep.
Then you can sequence it, but only in that order. You can't magically conjure something that you call a sequence.
So I'm kind of wondering then, and I'm going to go off on a little tangent here, but I think it'll help to focus the discussion. So bear with me for a second.
Okay.
We have this belief that there's a Wuhan lab of virology in Wuhan, China. Do you believe there's a Wuhan Institute of Virology?
Do I think there's a building over in China? I haven't been there.
Well, you've heard about the Wuhan Institute of Virology.
I have heard a lot of things.
Okay. So you even doubt that there's a Wuhan Institute of Virology?
I said that I am not going to sit here and affirm the existence of something that I haven't looked into.
Okay. All right. Well, Now, bear with me for a second, then.
You bet.
Let's assume that there's a Wuhan Institute of Virology, and that
Okay, there's a building that says WIV. Okay,
correct. Yeah. Yep.
Okay.
We suppose we believe Jon Stewart. For example, I mean, he did this wonderful.
Yes.
Did you see that?
He let us all engage in the wonderful establishment false dichotomy of wet market versus lab leak. Yes.
Yeah. Well, he talked about the business card and all this, but
it was hilarious, and it was very good at injecting false things into our minds.
Great. Okay. So under we'll call it the Steve theory, or the maybe we shouldn't personalize it. We'll just say the alternate hypothesis. The alternate hypothesis says that the Wuhan Institute of Virology was being paid to do gain-of-function research on viruses. Do you believe that there was anybody doing gain-of-function research?
On what?
On viruses.
No, they don't exist.
Great. So in other words, the U.S. government was spending tons of money funding research on a virus which doesn't exist. Coronaviruses don't exist.
The U.S. government was spending tons of money on stuff they're not telling us about.
Okay. So it's in other words, the virus is just a cover for a biological weapon or something else that we don't, but it couldn't be a virus because viruses don't exist.
Yeah. I mean, I'm not going to commit to whatever narrative you decide to write out of viruses not existing.
No, I'm not trying to,
viruses don't exist. Therefore, there are certain things that I conclude as a result are not true.
Okay. So it seemed to me, as a lay person looking at what happened, that the hypothesis that there is a Wuhan, that viruses do exist, that there was a Wuhan, or is a Wuhan Institute of Virology, and that the virus escaped from that lab and started infecting people because we had people that got infected, more people got infected, more people got infected, more people got infected. And there are noted virologists that then sequenced that RNA inside the capsid of that virus and published it.
They never found it inside the capsid.
Wait. So you're saying that the RNA that they sequenced was not from the capsid?
Yeah, it would actually be more accurate to refer to it as assembling a genome. They don't actually find an intact virion with an intact capsid. That never happens.
Right. Correct. This never happens. Mm-hmm. Right. Good. They assemble it.
They assemble it using, I believe they eliminate all base pair contigs longer than 150 base pairs. And then the computer begins permuting various potential genomes, comparing them to other hypothetical virus genomes in a database. And whenever they get to a percentage match of above 90%, they say, bam, there's our genome. It's it's purely on the computer. It's basically imaginary.
Well, it's not necessarily imaginary. It's just the way that they have to do it.
The way they have to do it requires them to create fake stuff.
Well, like they don't have the technology to take an RNA strand that that is 28,000 plus base pairs.
I thought it was 29,811.
Oh, sorry.
No problem. I'm sorry. I just wanted to make sure that we got that right.
Yeah. Let me see. 20. It was 28 or 29. So let me see.
Okay.
Yeah.
I mean, it's irrelevant. I just wanted to make sure I got it right.
Yeah. No. You're right. 29. Sorry, my mistake. I got the 8 and the 9.
Gotcha.
Okay. Yeah. 29,811. But again, the length of that will depend on the particular RNA, but somewhere in that order. So again, yeah. So these are assembled.
Yeah, they're assembled from a soup. A lot of the contigs have completely unknown provenance. There's going to be fetal calf serum. There's going to be nutrient medium. And then, so if you're assembling something from that, you don't know where these 150 base pair contigs came from. A great many of them have been shown to come from epithelial tissue from the lungs. So if you have 150 base pair contigs that come from the human being and not from the virus, then how exactly do you know where this genetic material came from? You don't, which makes the entire process unscientific.
All right, great. So if that was not a flawed process, and we could go and sequence substantially the same sequence in multiple people who didn't have the sequence before, and then they were in the same room or household with an infected person, and now that person now has that same sequence that we claim to be from the inside the capsid of SARS-CoV-2, would you believe that the virus exists?
No.
Wow.
I wouldn't. The work of Barbara McClintock has shown that the genetic material coming out of human beings from day to day isn't necessarily stable. Furthermore, the entire
yeah, of course it's not. I said, but if it's the same,
this only works if you find an intact virion. You have to sequence that. You don't get to take a soup of RNA and start piecing it together and saying, well, that used to be the virus. That doesn't make any sense.
But okay, so your claim is
not making any claims.
Okay, but what you're saying is that because we can't, so there's a strand of RNA, which if you pull, it's a three-dimensional thing, right? If you pulled it apart and you sequenced the 29,800 base pairs, and if you could do that without tearing it apart and then having to assemble it because you don't know what the shit you're putting in, if we could do that, then that would be convincing to you. If we could get a match between a person who was infected and then another person who then got infected from that person.
So I just want to make sure I understand. You find the intact genome, and then you look at each base pair, and then you find that it equals this 29,811 base pairs intact already, not sliced up into a million different contigs. And then you compare said genome to another person with an intact set of virions discovered, and it has the same person, and you do that with a large sample, and then I'll be convinced that you at least have a working hypothesis, but you still haven't shown that it caused the disease.
No, but if that person got sick, that should be proof, right?
No, it's not proof. You have to take the identified particle now and make somebody else sick with it.
Yes, but that's what I said you do. You basically, you want to take the particle and infect someone with the particle as opposed to the human being who is sick, then infecting another human being.
Okay, what do you mean infecting another human being?
I mean that if, like, my wife got sick from COVID.
Okay.
All right. And I'm in the same.
Okay. Your wife got sick
from COVID, right?
Okay. Well, remember, from the paradigm that I'm coming from, your wife got sick. I know
she didn't get sick. In your paradigm, I'm just saying in my paradigm. I'm just
fair enough. But for the sake of this discussion, your wife got sick. That's what we observed.
Correct. We observed my wife got sick. We observed that my wife tested positive on an antigen test several times, and that she finally cleared it. And then I got sick, and I started testing positive on an antigen test, and my fever went up, and I felt like this is not a normal cold, and I then started treating it. But the point is that I then had multiple antigen tests in order to see the progress of the virus and
your wife got sick, then you got sick.
The alleged virus.
Your wife got sick, then you got sick.
My wife got sick, and then
you
5 days later, I got sick.
You observe sickness occurring in close by in time and place.
Correct. Yes, so temporal. Yeah,
I'm with that. Okay.
Okay, great. So what was it that I caught from my wife?
I don't know.
No hypothesis on that one.
Well, I mean, I could come up with hypotheses all day, but that's not what this discussion is about. It is
but I'm just kind of curious, because if we could end this. If you have an alternate hypothesis, if you can tell me why all these people in Wuhan died, if you can tell me why 1 million people, or some order of magnitude of that, like 100,000 people supposedly died from COVID. If you can tell me what these people actually died from, Patrick, I would, the world will beat a path to your door. What is the alternate hypothesis?
Well, okay. I don't know whether the world's going to beat a path to my door, but I'll say this. Let's suppose that somebody gets murdered in Sacramento. Just bear with me on this analogy, okay?
Sure. Yeah, you bear with me.
Let's suppose somebody gets murdered in Sacramento.
Okay.
At the time of the murder, I was in Maine. The cops arrest me for the murder. I say, this is a video of me with 15 other people in Maine at the time of the murder, and they're all testifying that I was with them. I wasn't there. I have an ironclad alibi. And the cops say in response, well, then who is the murderer? And if you don't tell me, then we're just going to have to charge you for the crime anyway.
No. Okay, but so let me answer your analogy. Okay. The analogy would be that you tell the cops her let's see, sorry, who was murdered? Was it
John Doe?
So John Doe. Okay, so you say that Fred Smith called me up and he said, I just murdered your wife, or I just murdered John Doe. And cops, why don't you check? And I have a recording of it, Mr. Cop. Why don't you check that? And if you check his cell phone records, you will see that his cell phone leads to the body. In which case the cops will say, okay, fine. I don't have to believe you on your video, but I have really good evidence on the other one that explains the murder.
Well, for the purposes of this discussion, the debate is about this. Do pathogenic viruses exist? That's it.
I know that. I know, but
science is about falsifying hypotheses. Now, once we get past this virus stuff and we've really put it to rest, I would be absolutely happy to talk to you about alternative explanations for people getting sick. But right now, we really need to debunk this virus thing, okay?
Well, Patrick, you could debunk this in a heartbeat for me, as well as lots of other people, if you have the alternative hypothesis to say that if O.J. Simpson didn't kill Nicole, tell us who did, because that would be a great defense for O.J. Simpson to say it was the other guy. And we never heard that in his defense. Now, so if you can tell me what causes my wife to get positive on an antigen test, okay, it's lateral flow assay, essentially, and I turn positive on another brand of lateral flow assay shortly after she does.
Okay, now we're conflating the antigen test with the illness.
Explain how that could happen.
I don't have to explain that. All I have to do is show that the current hypothesis is unscientific.
But you're correct. You're correct. But the easiest way to do that, Patrick, is to explain to me.
I'm not going to give you an alternative theory to attack when the current theory on the table is unsupportable. Does that make sense?
Yes, it does. But as I said, Patrick, you can play it that way. But the easiest way to end the debate and convince me and lots of people is simply to explain the evidence in plain sight. Did she turn positive on a lateral flow test and then I turn positive a few days later? How does that happen? And in fact, I could have done 4 different brands of antigen test and they probably all would have been positive as well. Now, it's not guaranteed. These things have false positives, false negatives, but in general, these antigen tests select out different antigens. Each of these antigen tests that you can buy at a pharmacy is going to look at a different antigen. And that antigen test was verified against the SARS-CoV-2 RNA genome.
The one that hasn't been found?
Yes, that you claim hasn't been found. But see, ATCC would disagree with you because they sell lots of SARS-CoV-2 RNA sequencing samples for you to go and run your assay against and validate your assays against the RNA sequence of SARS-CoV-2. And different people, different companies will select different parts. And so if you got 3 different tests and they were all positive for my wife and me, then it's pretty hard to believe that she had virus A, which doesn't exist, and I got virus B, which doesn't exist. And it's just remarkable that these tests show and are the problem is, Patrick, that these tests are consistent with what we are being told by virologists. The behaviors, the observations are all consistent with what the virologists tell us. There doesn't seem to be any obvious inconsistency in terms of the observed facts. And so this is why people find it very difficult to believe the alternative hypothesis which you are proposing, that virology, which has been around for over 100 years, doesn't exist.
Okay, well, that's not what we refer to as an alternative hypothesis in the scientific method. I'm arguing for the null hypothesis. Now, another point that I'd like to make is what you're basically engaging in is a fallacy. It's called the affirming the consequent fallacy.
No, I'm not trying to do that. I'm not trying to say if you don't come up with the hypothesis that you're full of shit. What I'm saying is
that I would convince lots of people.
If you did come up with an alternative hypothesis to say it wasn't OJ that did it, it was Mr. X that pulled that strangled Nicole, or what, it was a long time ago. I forget how Nicole died, but all you have to do is show me that other thing and then show me the evidence for that, and we're done. We have to want to get into any of this stuff. Why can't you do that?
I can.
You can. Okay, great. What is your best hypothesis?
We're not playing this game. I offer no magical explanation for what people get sick with. That's not what I'm here for. I am here to critique the virus hypothesis. And we can go back and forth all day where you telling me that I'll have a pile of money heaped at my door if I come up with the answer, this, that, and the other. When it comes to figuring out why people get sick, that is an entire field all on its own. And I have my own theories, but that's all they are. Just
I'd love to, but can you give me your best theory, Patrick, for what happened there?
Not for the purposes of this discussion. It will only muddy the waters, and it will take us away from the topic at hand, which is.
No, but Patrick, I'll agree. You're going to give me such a great explanation for what happened. I'll agree with you like instantly. You'll say, Steve, she had this bacteria, or she had this fungus, or she had this, whatever. Just tell me what it is that she had that I got, and they both show positive on a lateral flow test that has been certified by the FDA.
Right. So you're engaging in a whole bunch of what we call a begging the question fallacy. You're saying, I got it from her. I don't know that. You're saying that the antigen tests for something very specific, an antigen that meant that you were fighting a specific pathogen. I don't know that. You're also begging the question there.
No. I'm asking you to assume, just assume that what I said is true. What I'm asking you for a plausible explanation of the observed fact that my wife had a positive antigen test, and then 5 days later, I had a positive antigen test as well, and both antigen tests were designed to detect SARS-CoV-2.
I don't know what
I'm asking you for a plausible explanation.
I don't know what they were designed to detect. That fact is not in evidence. There is no existent virus called SARS-CoV-2 in evidence. All we have is 2 people getting sick, one right after the other.
Yes, I'm just asking you for a plausible explanation. And
you're not going to get me to speculate during this conversation.
No, why?
because it would muddy the issue. The topic at hand is the
But money,
virus.
the issue, it's it's
It's
okay.
the virus
But
hypothesis,
Patrick,
which is
I
not
would,
scientific.
it won't muddy the issue. It will cut to the heart of this. We will be done. You'll tell me your hypothesis for what might have happened. And by the way, we can assume that these antigen tests wouldn't be on the market unless they were certified by the FDA, who has to go and validate those tests against against a sample of known coronavirus
That's
in
not
order
true.
to
That is not
certify
true.
those
The
tests.
FDA has never validated anything against a physical sample of a known coronavirus. That has never happened. And given the malfeasance of the FDA and their collusion with the pharmaceutical companies, I think we can agree that the FDA can't be counted on to do anything.
Yeah, but Patrick, we can validate this ourselves personally in our own homes from people who are saying, I got sick, I feel like this, I feel like shit, I lost my taste. My wife lost her taste when she got sick. The coronavirus is known. It's one of the few things I've never heard of another virus which causes you to lose your taste like this. And this thing is going around. And Patrick, here's the other thing, that she was golfing with a golf buddy and sitting in a golf cart, sharing a golf cart with someone who was infected. And she says, she told me that she tested positive and now I'm feeling sick. And then my wife suddenly. So it's not just two of us. It's a chain of three. It's her friend. And I don't even know what her friend, she probably has her own story, but her friend got sick, tested positive in the antigen test. My wife then got sick, tested positive on the antigen test. Now I got sick and tested positive on the antigen test. What is the most likely explanation of how that can happen if the virus doesn't exist? Speculate for me.
No. Yeah, you know what? I'll give you something. So I go to the beach and I get and all of a sudden my skin turns red after a couple of hours at the beach. It turns real red and skin starts flaking off. Then you join me at the beach. And then a couple of hours later, your skin starts getting really red and it starts flaking off. And then you point at me and you say, you gave me a virus. And then we go to the doctor and they take little scrapings of our skin and they find dead skin tissue on both of us. We test positive. We test positive for the dreaded sunburn disease. And you, because it's a similar observation, believe that I gave you the sunburn disease.
Yes, but Patrick, so to use your analogy then, which I think it's a great analogy, Patrick, but you see, if somebody was saying, hey, I'm a virus denier, they would, and I said, but this sunburn thing or this skin disease that we both got when I met you at the beach, you would then immediately say, Steve, you're full of shit. It's not a virus at all. It's caused by the sun. And that's the alternate hypothesis. And then when you said that, I would immediately say to you, oh my gosh, how could I be so dumb? You're absolutely right. That is the better explanation of it. That would fit the facts. It's consistent with the facts. And
you're very insistent on that I speculate for you. It seems very important to you.
Oh, it is. It is
Okay.
because we could end this. We don't have to get into sequencing. We don't have to get into Sanger sequencing. We don't have to get into
if I give you an alternative explanation for why your wife and you both got sick close together,
her friend got sick too.
Mm-hmm.
And then she said, Michelle, hey, I wanted to let you know we were in the golf cart together. Mm-hmm. I just turned positive on my COVID test. You should probably, watch out. My wife was saying, hey, great. She wasn't feeling sick. Then like 2 days later, she was sick. And I said, maybe you should take a COVID test. Right. Mm-hmm. So she got sick from her friend had a positive antigen test. My wife got a positive antigen test. And then I got a positive antigen test. And they're all temporally sequenced. So explain how that happens to me. You just give me your hypothesis. It doesn't have to be right. Just give me a plausible hypothesis. That's all I'm asking.
I don't have one.
It's like the sunburn.
I don't have one, Steve. I'm so sorry. I'll go work on it and I'll come back to you. Okay.
Okay, great. Because that would facilitate things. Great.
Okay.
Thank you.
No problem.
Okay, great. So, so we can get back to the our list here. Find it in fluid or tissues, extract the fluid, done that. Okay. Extract a sample of the particle, not done that. Only the particle. So in an ultracentrifuge, I said you can't extract only the particle in an ultracentrifuge. It's not capable of that. You'd agree with that, right?
They were able to do it with bacteriophages.
Bacteria. Okay. They're able to just isolate that one bacteria.
No, I mean, a sample thereof of only that particle. They were able to do it with bacteriophages, which are of similar size and morphology to the hypothetical virus. So they could have done it if such a thing existed.
They could have done it. Well, so you're saying that you can isolate a single bacteria?
Nope, I am not saying that. I am saying you can isolate a sample containing only that particle. Many examples of such particles.
Well, but bacteria is much easier to isolate, right? Because bacteria live on their own. They're large.
I wasn't talking about bacteria. I was talking about bacteriophages.
Okay, what's the difference?
A bacteriophage is a very, very, very tiny living part, something that comes out of a bacterium. And they have been shown, characterized, observed, studied, and they definitely exist. They've been isolated, they've been purified, and they are also of the same size, shape, the rest of it, characteristics, basically, as a virus, which is hypothetical. Has never been isolated using the same method. So okay. Yeah, it could be done.
Okay, can you do it for excuse me, and so you can. So I'm not familiar with that, so I'm going to have to take your word for it.
Okay, they've done it with. They did it with sucrose gradient, and they also did it with density gradient ultracentrifugation.
Okay. Again, I can't comment on that. I don't have that background. I do a lot of stuff on my Substack. I'm not an expert in all this area, but so
that's why this is just a question and answer session. I'm sure
correct.
People.
Correct. Yep. So your point is that you can do it for. So let me ask you theoretically. Okay. So these virus particles that people claim exist. These virus particles are very small, and there could be lots of them. I could be having three different. I probably do have several different variants of when I had SARS-CoV-2. I probably had different variants of SARS-CoV-2, but there's a lot of. But I could have three different viruses, or two different viruses. I could be infected with multiple things. So. I guess if you got somebody who is only, was perfectly healthy and then got infected with the SARS-CoV-2 virus, is the claim that the only particle of that size and weight that would be extracted in the centrifuge would only be a viral particle?
Well, let's take your example. I'll sit and beg the question with you, okay?
Okay.
What you want to do is if you want to discover a pathogen, you got to find a whole bunch of people all exhibiting the same disease. Then you take samples from all of them. Then you do, for example, a density gradient ultracentrifugation. And then you look at all the bands and you see if you can find a band in the centrifuge. You can find a band that's common to all of those people exhibiting the same disease. And then you compare that band that's common to all of them. And then you have a working hypothesis as to what got them sick. There could be multiple different such morphologically similar particles in different people. But you find the one that's in common, and then you hypothesize that that's the cause of the disease.
Okay. Now, has that been done?
No.
Really? In 100 years, that has not been done?
Yeah, it's really, in virology, it's never been done.
So has someone done the reverse experiment to show that you don't get anything, that there's no commonality?
Yeah, it's been a whole bunch of failed experiments.
Wait a minute. Failed experiments. By failed experiments, meaning that
No
when you looked at the
hypothesis
density
constantly affirmed.
gradient, that there was no commonality between two people in the same household who got COVID essentially from each other, that there is no commonality in the density gradient of doing a centrifuge to separate out the virus.
That has never happened, and I know it's shocking.
Wait a minute. When you say it's never happened, do you say that nobody has actually done the experiment, or people have done the experiment and it failed?
Oh,
So that's
gosh.
two different things.
Well, virologists don't bother with a centrifuge. They do what we call a cell culture, which automatically is invalid. So the cell culture is a totally different topic, but and essentially they conjure what they call the pathogen from that. And then by the way, they don't bother trying to see if it gets anybody sick, but they conjure a particle, and then they just show you the electron micrograph, and they put a little arrow, whatever schmutz comes out of the cell. But no, they've never done any kind of ultracentrifugation to isolate a particle that could be hypothesized to cause illness in any sick person.
Okay, so they basically have never done that experiment. Now, suppose that they did do that experiment, right? And it showed that the bands essentially lined up.
You mean if they had done something they never did?
If they actually had done the experiment, they have two family members, one, clear or hypothetically, on the surface, Occam's razor suggests that person A got, infected person B under the viral hypothesis. So then you go and do your centrifuge, and then you compare the results. You said that's never been done.
Correct.
Okay, so if we did that, and the centrifuge results lined up with each other such that these people seem to have a viral particle that's the same density and so forth as the other person. Mm-hmm. Oh, and we scanned them before when they were perfectly healthy, right? So
now they're sick.
And now, so we did a reference scan, right? We did the control, right? The control is that they're not sick. Okay, then we come in with the bag of coronavirus. We open the bag and we infect them. I mean, you can't do that ethically, okay? But suppose we could, we could do it. I guess we could
animals.
We could do it in animals.
Mm-hmm.
Okay, that'd be the way to do it, right?
I'd take it. Yeah. So basically, what you're saying is, if the scientific method were observed and it showed the existence of pathogenic viruses, would I believe in the existence of pathogenic viruses? Sure, I'm a believer in the scientific method.
Great. Why don't we just do that experiment?
It's never been done, and it never will be done. And your virologist friends will explain to you why they can't do it.
Why can't they do it?
Because the viruses have never been shown to exist. And the scientific
No. I'm asking why the virologist would tell me why can't I go to a virologist and say, do this experiment?
Do that, find out that there's no viruses. And the ones who go public with that get all their funding taken away from them.
Yeah, but man, I know there are some renegade scientists. We call them, red pill scientists that would love to do something like this and put a feather in their cap.
Stefan Lanka? Stefan Lanka did exactly what you would expect. He proved that the cell culture is the experimental procedure is what causes the cytopathic effect. And I know we haven't gotten to the cytopathic effect just yet, and we really needed to, but it's such a long discussion. And I think some of the audience gets it, but it requires a little bit of background to really understand it. But you find sick people and you centrifuge their fluid. If you do that, the experiment will fail. And your virologist friends will probably explain to you why the experiment will fail in their own terms. And what they say, and I can even tell you what they say. They say that there's not enough virus to find. You can't find the virus in a sick person because there's not enough of it. But the way that viruses make you sick, hypothetically, is that they invade intracellularly. They get into the cell, they hijack the machinery of the cell, and they cause the cell to produce an arbitrarily large number of copies, causing the cell to die and rupture. Out come the more viruses, repeating that cycle over and over again until you're sick. Now, here's the problem. Let's suppose you lose, say, a pinhead size amount of your lung tissue. You'll never notice it. If you were to lose a golf ball sized amount of your lung tissue, you'd have a bad day. A baseball sized amount of your lung tissue, you'd have a really bad day. But if it's affecting that much of your body, to argue that there's not enough virus in there to find under any circumstances?
Well, people have different express different viral loads when they get sick.
They don't actually find any viruses. So how exactly do they have viral loads?
Well, again, we're assuming. Excuse me. We're assuming for the purposes of this discussion, I'm taking the side of the virologist, and the virologist would claim that, of course, a virus exists, and they would claim that different people have different viral loads when they are infected. And we can see this in a quantitative PCR test where...
Do you know how the PCR test works?
I'm not an expert on how the PCR test works, but let's not go there for a second. Let's just... I mean, because you're claiming that the virus doesn't exist and therefore there are not different viral loads, but under their theory, okay, when we don't have to talk even about the PCR test, but under the theory of virology, and because we're trying to poke holes, we're trying to find the self-consistency error in virology, okay? And the virologists claim that their theory is that when people get infected, that they have different viral loads. And in fact, the papers that I've seen showed that the higher the viral load, the greater the infectivity. So you're more likely to get someone sick if you have a higher viral load than if you have a lower viral load, which again seems pretty intuitive and more importantly, it's self-consistent. So when they say that you may not have in these sick people who may just get a mild cough or whatever, that there may not be enough viral copies to succeed in the centrifuge example, why would, I agree, if these things are causing massive lung injury and these things are found in every single cell of your body, that this virus has invaded and attacked every single cell of your body, I'd agree with you that, you should be able to find that in a centrifuge.
Okay, well, and how do you feel about the fact that it has literally never been found in a centrifuge?
I don't know. I don't know enough about the centrifuge technology to be able to give you an intelligent answer to that question. And so I would love to answer that question. I don't have the domain expertise to answer that question. And I'm not the kind of a guy who, makes stuff up. So I will just admit that I don't know the answer to that because I've never done a centrifuge. Yeah, I mean, the whole issue here is that the methods used by the scientists are unfortunately, this is not a perfect thing where we can control everything. So shortcuts.
The scientific method requires rigorous controls, and whether or not, if a scientist comes and says, well, the scientific method it's just obsolete in virology, and they kind of do say that when you really pin them against the wall, that's not scientific. So what they're doing is, in my opinion, is scientifically flawed. It doesn't adhere to the scientific method, which is very, very well defined. The scientific method is you start with, you observe a natural phenomenon. Somebody gets sick.
Yeah.
Then you research, you observe further in order to form hypotheses. One of the ways that you form hypotheses is through epidemiological observation. Your wife got sick, her friend got sick, then you got sick. Say, wow, we all got sick in the same time and place. Whatever could it be? So then you start looking at your tissues and you try to find something similar among all of them, whether it be electrophoresis, sucrose gradient, whether it be ultracentrifugation, whatever. Then you test that hypothesis under very, very strict controls. And then you have to constantly figure out, what could I be doing wrong? What possible cause of this could I be completely overlooking? And you do it over and over again until one day, whatever your conclusion is, gets falsified by somebody else because there was something you didn't think of. It's a rigorous process, and nothing like that's going on in these viral experiments.
Let's put it this way. I haven't seen any evidence. Now I'm willing to be convinced, and presumably you are as well, that
Always.
Good. Okay. So similarly, there is the vaccines don't exist hypothesis, viruses don't exist and viruses exist. And there's a bunch of evidence coming in. And I would then take that evidence coming in and say, is that evidence consistent with the virus, that there is a virus, or is that evidence consistent with there isn't a virus? And so I would be looking at lots of different things. And I would say, in 100 years, Stefan Lanka is the only guy. It was the first guy in history. Like, when did this, like, is Stefan Lanka the first guy to call fraud on virology? Or has this been going on for decades?
Well, I mean, if you look at Enders' paper in 1954, when he supposedly isolated the measles virus, even he admitted that the control experiment also caused the cytopathic effect. And he said, well, we better look into that. And then he won the Nobel Prize for isolating the measles virus. And then we were off to the races from there. So, yeah, I mean, basically, if you look at Enders,
I'm asking, who is the first guy to say the emperor has no clothes?
Is this like bar trivia? I'm not sure. I know that.
No, I'm not trying to be cute here. I'm just
Stefan Lanka proved that the cell culture shows the existence of nothing causing. Stefan Lanka
lost his 100K euro bet.
I bet you the New York Times also said that he was wrong. I bet you there's lots and lots of establishment.
No, but a court of law where they had experts and a judge to evaluate what the expert testimony was said that Lanka lost his bet on whether the measles virus exists.
The opinion of any court is of no value when it comes to scientific questions. How did the experiment go? It proved conclusively the cell culture is invalid.
So, and by the way, it's 3:00, so I have to end this.
Thank you very much for a spirited.
But hang on. Yeah, I just wanted to just finish that last thought before we do that, and thank you as well, because I'm trying to learn this stuff as well and get this laid out so that we can have a very efficient discussion for debating these points. So this has been, I think, helpful, and I'd like to get a copy of this so I can send it to my virology friends so they can take a look at this. And
it's uploaded to Rumble right now, so you can just grab it that way.
Okay, yeah, if you can just send me the URL.
We'll shoot you the link immediately after we're done.
Yeah, okay, good. But to that last point about the fraud, so my question was the difference. So with vaccines, there were lots of people calling the CDC out on it very early on about thimerosal. So Andrew Wakefield, was saying, "Hey, emperor has no clothes." Bobby Kennedy, emperor has no clothes. More and more people. How many? So it seems number one, it seems very odd to me that we can go 58 years and nobody not one person is speaking out about the fraud. And the second thing is, how many virologists today support Lanka's view that virology is a fraud?
You mean how many people in virology who make money from virology would like
No, I didn't say that. No, I said how many virologists, because there are a lot of virologists who are retired. They have no more skin in the game. They can say anything they damn well please because they're done. They're retired. And some of them are interested in the truth. It's the same thing for the vaccine. When we have retired doctors, they're not afraid to speak out. But anybody in the profession right now gets deplatformed if they speak out. So I'm not necessarily saying, find me a working virologist. I mean, that would be impressive. But How many total virologists, since Lenka came out and said the emperor has no clothes, how many total virologists, probably retired, but in 10 years, that's a lot of retirements of virologists who would then go and say, Lenka was right. Let's do the right thing. I'm going to be on the right side of history and I'm supporting Lenka. How many virologists have done that?
I have no idea. It's probably a tiny number. One of the biggest things that Mark Twain ever said was, it's easy to fool somebody, but it's way harder to convince them they've been fooled. And if you've spent your entire life as a virologist, think about the cognitive pain that would result, that you have been essentially engaging in a decades-long malicious fraud.
I get that argument, Patrick, but you see, let me give you another analogy for you to consider, which is for the vaccines. What would it take for people who were supporting the vaccines for all this time to then come out against what they were told? And the answer is, it's more than one guy. Okay. It's a lot of guys. It is, in the case of the signing the Great Barrington Declaration, or these, the doctors, there's 17,000 doctors that are speaking out against the vaccines that they were told is safe and effective. 17,000. That's a lot of people. They're very high profile, very credible doctors like Peter McCullough, for example, and Robert Malone, who are very credible scientists who are speaking out and risking all of this retribution. And it's not just a couple of guys that have stellar reputations. It's a lot of people that are doing this. And the list grows longer and longer every day. Now, Stefan Lenka has had 10 years. Sorry, he said 12 years, or more than 10 years, to accumulate a following from when he said, hey, there's the emperor has no clothes, and yet you don't see this pickup of, oh, there's another virologist agreed, another one. Whereas with the vaccines, we got a lot of people who basically risked their careers and risked their livelihoods and risked their income stream, like Brett Weinstein, for example, who basically says, yes, these vaccines are not safe. He was the one who gave us the initial platform on this. And since then, lots and lots of people have been convinced. Now, I grant you that there are a lot of people who are lay people who subscribe to the, I don't believe there's a virus. I believe the people who claim that there isn't a virus. There are a lot of people that do that. It's not 1%. It's pretty substantial. I did here, let me just pull this up real quick. This is a poll. It's on my Gab account right now. And the numbers are 52% says, yes, the virus exists, many strange. 32% believe it's a hoax or bad science. So that suggests that there are a substantial number of people who believe what you're saying. Okay. But the fact that you don't have the professional virologists, like, in analogous to our, vaccine. I mean, I guess you could say, oh, well, how many infectious disease professors do you say that, are coming out saying that the vaccines are unsafe? And I would say, very few, right? So I could make that analogy, but, there are enough of these people that are highly credible. There are multiple very highly credentialed people coming out, very highly respected people coming out saying the vaccines are unsafe. Why hasn't that happened with the virus?
I got an explanation for that.
Okay.
The fraud that is the medical industry in general is built on premises that stack on top of each other. And what we're finding out as we dig through is that it's not just the top couple of layers that are fraudulent. And once people get to this vaccine is toxic, they start looking at other vaccines. And then once they kind of break through oh, wow, they really are just toxic, then they go down to the underlying premise. And we're not quite there yet. Only a very tiny group of people are in the category of, there are no pathogenic viruses. It was a fraud from the beginning. Now, I'll tell you a quick anecdote about myself. when this entire information war really kind of broke out into the open, in my opinion, in 2017, I noticed that institutions were corrupt. I noticed it earlier than most, I think. And one of the things I said to myself probably by late 2017 is, I'm not going to accept the opinion of any expert on anything without checking it out myself down to the most granular level that my mind allows me to. And every single time that I have gone through that process, never trusting an expert, no matter who they are, I have found, oh, wow, this is a fraud too. So that's where I am with virology, and I'm very confident in the conclusions that I've drawn.
Okay, good. Good. This is really interesting because, see, I'm the same way as you are, right? I don't trust these people either. I trust them as far as I can throw them, which is not very far. And so I don't take, like people told me safe and effective, safe and effective. And I, bought that, it wasn't until May of this year not this year, but May of 2021, when I realized that I had been had. And since then, it's like my doctors tell me, my cardiologist says, you should be on statins. And all of my friends who are smart tell me, avoid the statins. Because yes, they'll lower this, but they have these really bad side effects like, I don't know, Alzheimer's or whatever that they're not telling you about. Okay. And I believe the people, my smart friends who are telling me this, and I'm not believing the cardiologists. Okay. So we're similar in that we have a distrust of people and so-called experts, and we want to see the data. Okay. So we're aligned on that. And, I love that about you. That's great. Okay. So in this particular instance, though, we're on opposite sides of the fence, because when I talk to my very smart friends who are red-pilled on the virus, to a T, to a man, they are all like, this virus does not exist, is junk, and they won't even spend the time. It's like they have the attitude on the virus is junk thing, exactly the same as the attitude of the pro-vaccine people who look at people like me and say, oh, this guy's not worth debating because he's just a nutcase who doesn't know what he's talking about. Now, it's interesting that my friends, who have tried to get to come to the debate, would say, I'm not even going to spend the time on this. This is so junk science that, and so I'm seeing the same sort of thing, right? The way that we're treated as misinformation spreaders is the same way that you're being treated as saying that, that the virus doesn't exist. So yes. What I'm trying to do is to go and plead with them to say, let's be better than the other guys who don't give these guys the time of day, but let's examine this, because clearly there are a bunch of people who believe this, and it's important to settle the issue. So I believe it's important to settle the issue. I think that Sam Bailey is not going about it the right way, because she should engage people like us who are in the middle and say, hey, we just want to know the truth here. And if I'm being misled on whether there is a virus or not, I want to know that, because I don't want to be spreading misinformation. I'm perfectly willing to hear all the evidence on this, and to make sure that my qualified friends, and beg and plead them to come to the debate table and let's settle this. So this, what we've done here, although it was kind of long, but I think it had to be, because we had a lot to cover. But I think this then now forms the basis of having a more productive discussion. And there may be, a second version of this that we have to do with someone who knows more about this than I do, where we can go into the nuances, and then we can have a full-blown debate. Is that fair?
Wherever I will do it anytime you want, and I will get out of bed if the time zone doesn't match up.
Awesome. Great. So let me see what I can do. And thank you for doing this, and thank you for your time here, educating me, putting this together, and hopefully we can find a resolution to this, because I don't have a horse in this race.
I'm really glad to hear that.
Yeah. I mean, look, I'm only saying that the virus exists because it's consistent with everything I see, and I don't see any evidence coming in to suggest an alternate hypothesis that is a better explanation for it. Because once I see a hypothesis that is a better explanation, this is why I kept on you on this. Once I see that hypothesis that's the better explanation for what is occurring, then you got my attention.
We'll get there. I think we
Okay.
will. You need to explore the virus hypothesis a little bit more deeply, and I think you might be kind of shocked.
I'm willing to. I'm here because I'm willing to listen and be convinced. I think that any legitimate scientist should be open to listening to people who don't agree with them and to evaluate their arguments and come to a conclusion. And right now, we need to explore, sort of further down and explore some of the issues that have been raised here. So we've just taken the first step. And, thank you for doing that with me. And, we'll see. At the end of this, either you're going to convince me or I'm going to convince you, or we're going to agree to disagree. Mm-hmm. I mean, those are the only options.
Yep. Very true.
So, yeah. Okay. So good. Well, thank you very much. I certainly appreciate it. This is super helpful. Yep.
Enjoy your trip to the airport.
Yep. Thank you.
Cheers.
All right. See ya.
All right, my dear friends, that is our show for the day. I hope it gave you food for thought. I hope it'll cause you to look into some of the stuff that we talked about today. It is, of course, my conclusion that virology has been a fraud from the very beginning. And I'm willing to defend that proposition, and I'm willing to defend it to anybody in the world. Big thanks to Steve Kirsch for having the moxie to come on the Reading Epic Threads show. And I think I'm going to go ahead and wrap this up. Don't forget, guys, go to ret.isogenics.com, ret.isogenics.com. And I created this dummy account just so I could be able to do this. ret.isogenics.com. Get your collagen elixir, get your nootropic elixir, get your isogenesis vitamin packs. I love you all. Let us pray. Saint Michael the Archangel, defend us in battle. Be our protection against the wickedness and snares of the devil. May God rebuke him, we humbly pray. And do thou, O Prince of the heavenly host, by the power of God, cast into hell Satan and all evil spirits who prowl about the world, seeking the ruin of souls. Amen. I love you all. Thank you so very much for being here. And I'll see you tomorrow.
I will be the greatest president that God ever created.